about the evolutionary developmental biology of flight, and in particular, the evolution of feathers. So as you might expect, it's thought that the evolution of flight and the genes that enabled birds to fly evolved somewhere in the lineage that descends from the archosaur ancestor, which is the common ancestor of crocodiles and birds. And so these sorts of changes in genes that enabled, for example, reduced body size, the hollow bones that are lightweight that allow birds to fly, and particularly feathers, those must have evolved on this branch. Or maybe not. So on the other hand, if the evolution of flight evolved somewhere farther in the past, that would indicate that each one of these separate lineages would have had to have lost the ability to fly independently. So it makes more sense from a parsimony perspective that it's a descendant of the last common ancestor of crocodiles and birds that had all of those changes occur in the DNA that allow birds to fly. So one of the things we'll have to talk about then is the evolution of feathers. And if we take the structure of the feather and break it down into the smaller components, what one of the evolutionary novelties is, perhaps, has to do with keratins. These are proteins, and there are essentially two different structures of keratins. They're the keratins known as alpha keratins, and they're called that because the proteins tend to adopt mostly an alpha helical structure. So when the amino acids in the primary sequence of the protein or the primary structure fold, they tend to fold into helices. On the other hand, there are also beta keratins, which tend to form a different secondary structure, beta pleated sheets, where the string of amino acids winds back and forth, making a sheet instead of a curl. And the amino acids then chemically pair or bond with each other to form a rigid sheet-like structure. So it's known that the alpha keratins tend to be involved in producing things like mammalian skin, nails, claws, and hooves, and our hair. Not that I have that much of it right now. Alpha helix proteins. And then the beta keratins that form these beta pleated sheets tend to be involved more in skin and in birds, scales, beaks, claws, and feathers. And you might be thinking, wait, do birds have scales? And they actually do. So scales and feathers are both comprised of keratins. That's not the only component, but that's one of the main components. So we're familiar with, I think, the idea that reptiles have scales, like snakes and lizards. Mammals also have scales. There's one mammal, the pangolin, that independently evolved scale production. So in the outside of its skin, it produces scales, and then bird feet tend to be scaled. So while we think of birds mostly as being feathered, they do also have scales, and birds also independently evolved that same beta keratin structure that helps them produce scales. So these are interesting traits to think about, the evolution both of scales, and today, when we talk about feathers, trying to understand what is the evolutionary basis of that developmental change. So what happened at the genetic level to allow these sorts of traits to evolve independently? So here are some of the big questions related to this topic. First, the biggest one, how did feathers evolve? And then more pointedly, are they related to scales? That is, does the same genetic toolkit, does the same set of genes that produce scales also produce feathers? And then what genes facilitated the evolution of feathers? When, in particular, related to that phylogenetic tree we just looked at, did those genes arise? Is it really true that all of the genes that are necessary for flight evolved just on the lineage of birds? Or maybe did some of those proteins and genes exist farther back in the past? One way to approach this is to take a set of genes from published research studies, not necessarily by the authors this week, that are known to be either expressed in feathers or necessary for birds to produce feathers. And this is a small part of the list from the same manuscript that we're looking at, the supplementary table 1. And this is just a tiny part. One of those genes, just as an example, is a gene called ectodysplasin, or EDA for short, EDA. And mutations in this in humans are known to cause phenotypes. And so that is just an example of one of these genes and how it's known to be involved in basically ectoderm development. In this case, in humans, it's skin, but maybe in birds and other animals, that gene is essential for feather development. That is, it's necessary for feather development. And once we have this list of candidate genes, genes that we might expect are useful or essential for feather development, we could then look at the sequence of the genes and the protein sequences that they encode and ask things like, in non-birds, like reptiles, and in birds, are these protein sequences conserved? That is, does the gene encode the same protein? Does the protein fold the same way? Does it probably have the same function? If that's the case, then that protein is probably not involved in feather development specifically. But if we see differences in where the gene is expressed during development or where in the body the gene is expressed, if there are sequence changes to the gene, those sorts of changes between, again, say reptiles and birds could be explanatory differences for why birds develop feathers and reptiles don't. And then in terms of how you would determine if there were feather-related genes when they showed up, in general, that's pretty straightforward. We already mentioned this, the idea that if there are genes or genetic changes, that is, whole new genes or changes to existing genes that are essential for feather formation, we would expect to find those changes, those mutations, again, either the production of new genes, say through gene duplication, or specific mutations to existing genes, we would expect to find those only in birds. In other words, if we looked at any bird and its DNA, we would see those changes. And as soon as we moved outside of birds, the descendants of the archosaurs, so crocodiles, for example, or lizards or turtles or mammals, no other species other than a bird would have those same changes. If, for example, we found those changes that are essential for feather development farther back in time, that is, those were shared by birds and crocodiles, say, then we would know that flight was possible, perhaps, in an ancestor, and for some reason, the descendants of the archosaurs that became crocodiles lost the ability to fly. Now, so far, I've been telling you a little bit of a half-truth, that sure, we do want to know when genetic changes occurred that allowed the production of feathers and allowed the evolution of flight. But the story's not just about genes that encode proteins and sequences inside the genes that could change, mutate, and cause the protein sequences to change and thus their function potentially to change. The gene is one part of the story, so what is the other part of the story? Well, the gene isn't enough. You can have a gene. You could have a gene that is necessary for feather production. So let's call it the feather gene. I'm just telling a story. So birds could have that gene, but that's not enough. That gene has to be transcribed and translated and produce a protein, or maybe just an RNA. It depends on what the feather gene does. But somehow that gene has to be expressed. At the very least, it has to be transcribed. And that means that it needs to have a promoter and probably one or more enhancers. And those are the sequences that regulatory proteins will bind to on the DNA and first facilitate transcription, the first step in the central dogma. So not only, in other words, do we need to worry about what's happening in terms of mutations in genes that may be involved in feather development, but also it's possible and perhaps likely, again, this is a major aspect of the concept of evolutionary development, that it's not necessarily the protein coding sequences that evolve or change or mutate to allow the evolution of novelty. It's very likely that a lot of the time, mutations in regulatory sequences that change where that gene is expressed, that change when during development the gene is expressed, for how long it's expressed, in what tissue it's expressed, and so forth. Those sorts of changes often seem to underlie evolutionary change. So we need to consider both of those types of changes. Changes to the coding sequence, that is the region of the gene that encodes protein. Those sequences are certainly important because the DNA sequence dictates the RNA sequence, which dictates the amino acid sequence and the structure and the function of the protein. But regulatory sequences themselves are important. So what happens if there's a mutation in a promoter or an enhancer that turns on or increases the expression of a feather-related gene in the developing upper limbs, arms, essentially, of birds, the things that would become wings? So again, the concept here, just to reiterate, is that we would want to sample multiple birds and then multiple non-birds, look at their DNA, compare the DNA sequences, and find mutations that were specific to different birds. So between birds and non-birds. So for example, we could have a chicken and a sparrow and a crocodile and a lizard, let's say. And we could look at the same sequence. This could be regulatory or protein coding. It doesn't really matter for this example. And we know that there's going to be some genetic variation. So I'm just going to pick the first nucleotide to indicate that we know that over long periods of time, random mutations naturally occur in genes. So all of these 4 species will have some spots when we line up the same gene and look at the same nucleotide in the same gene. There are some spots where they will be different. There will be other spots where the nucleotide sequence tells us something about the ancestry. So here, the crocodiles and lizards that diverged from birds farther in the past share a C, and all of the extant birds, the birds that live today and all the species that we're looking at, have a different mutation. So that would be the sort of change we would look like for a possible flight-specific mutation. It doesn't mean it is, but it means it's a candidate mutation because it is present in all flying species and not present in all non-flying species. And then, as is often the case, most of the nucleotides in the 2 gene sequences would be the same. Just for completeness. So when we compare DNA sequences from vastly diverse species, even individuals from the same species, you will see some mutations that are indicative of the relationship between the species. The fact that these 2 individuals share an A at that position is because they're both birds. They're closer related to each other than they are to crocodiles and lizards. And so that's why the crocodiles and lizards, say, have an ancestral nucleotide. So in the ancestor, there was a C, but in the ancestor of all birds, there's a mutation from C to A, and that's why the descendants of that ancestor, all birds, share that A in this example. So that's essentially how we would identify and get a date on the origin of these sequences. We'd look at more and more distantly related organisms and find out when we no longer saw that shared mutation. And that would tell us where in time the common ancestor where that mutation arose. Along the same line, there's an important concept for identifying regulatory sequences. It's called phylogenetic footprint. And we're going to look at how to produce a phylogenetic footprint just briefly. And the concept here is in regions of the genome, so let's look again at an example where we're looking at a chromosome sequence of birds. I'm just not writing down the nucleotide sequence, and the same region in, let's say, a crocodile. And let's pick a particular bird, we'll just say chicken. So single species. So we're looking at a chicken chromosome in the same spot, in the same chromosome in a crocodile. We would expect to see in the region of genes, so let's say that there are 2 transcription start sites in this part of the chromosome, and that those encode, those are the start of the locations of genes. So we know where on the chromosome the gene is. But remember, the gene where the protein is encoded is just one of the types of sequences that we're interested in assessing or analyzing. The other one are those regulatory elements. And regulatory elements are unlike genes in that the sequences of genes tend to stay, as we just looked at, mostly the same. So there are going to be rare nucleotide differences like that T to A, but in general, protein sequences will remain the same, or almost the same, so that we can identify the genes, because genes tend to be more sequence similar to each other from even just different species. That's not true usually of DNA that's non-gene. So extragenic DNA, because it doesn't always have a functional role, natural selection doesn't necessarily resist mutations in those parts of the chromosome. So this is where we'll often find totally different, random, essentially, sequences between very distantly related species. This isn't true. So the closer two individuals are to each other in relationship, the more similar their DNA sequences are. Human chromosome sequences are about 99.9% similar to each other. But if we looked at humans and chimpanzees, it's about 99%. If we look more distantly in the past to find a common ancestor, that sequence similarity drops off precipitously. So if we're looking for regulatory DNA sequences to compare, to again ask that question of which individuals from which species share common mutations that might be involved in feather development, we need to find where those regulatory sequences are. So in blue, I'm going to suggest that this is a region of an enhancer. Now, an enhancer doesn't encode a protein, but the sequence of an enhancer is very specific usually. So there might be, for example, a protein, a transcription factor, that binds the enhancer sequence TATG. That means that mutations to that enhancer sequence would have a fitness consequence. If the mutation happens there, that would cause the transcription factor no longer to be able to bind the sequence, and so it would no longer be able to activate transcription of a gene. So if this first gene was the feather gene, let's say that here the enhancer sequence in the crocodile is TATA. If TATG is important for transcription factor binding and the initiation of transcription of the feather gene, that works in birds, which we would expect because birds have feathers. And this mutation in an enhancer might explain, for example, in this hypothetical situation, why the feather gene is not transcribed in crocodiles that don't have feathers because of this mutation in a regulatory sequence. But notice that most of the regulatory sequence is conserved. So in non-genic DNA, DNA outside of the coding region of genes, sequences tend to degenerate or change rapidly. So there's not much similarity between the non-gene sequences of different species. Here's where the phylogenetic footprinting comes in. If you compare these two sequences, we do find that the regions of similarity, there's similarity between those sequences where there's a gene and those sequences where there's a gene, And these sequences, except for that one point mutation, the enhancers also tend to be conserved sequences, but they're not genes. So this is how enhancers can be identified, and that's what phylogenetic footprinting is. Simple. I've already drawn it here. You line up the sequences of two chromosomes, the same part of the same chromosome, and you look for regions of the chromosome that have similar sequences that are not in genes. And those are often used to produce, those alignments are used to produce percent identity plots. This is also called a VISTA plot. We've seen these in previous videos, unless this is the first video you're watching, in which case, welcome. The VISTA plot on its y-axis tells us the percent similarity in DNA sequence between two different sequences. Seqs, S-E-Q-S, for sequences. In this case, this is a percent identity plot between an X chromosome and a Y chromosome. And as it says, these coordinates on the bottom are X chromosome sequence. So we don't actually see the nucleotide sequence of the chromosome, but we see that every one of these tick marks represents 1,000 base pairs of DNA on this chromosome. So that's the X chromosome sequence. And these shaded boxes or plots represent for each tiny little section of sequence, in this case, every 100 base pairs. So for the first 100 base pairs, for those 100 base pairs, we've aligned the X chromosome sequence with the Y chromosome sequence, the homologous chromosome, and asking over those 100 base pairs, how many of the nucleotides are the same? So you can imagine if there's one point mutation in those 100 base pairs, that would produce 99% similarity in that region, and we would plot a dot at 99%. And then you move to the next 100 base pairs and you ask, in that next 100 base pairs, what's the percent similarity? And you move to the next one and the next one. So you slide along the X chromosome in 100 base pair windows, 100 base pairs at a time, and you calculate the percent similarity between the X sequence and the Y sequence. So we can see that, for the most part, the X and the Y sequence are close to 100% identical. Notice that the scale only goes down to 50% in this plot. But there are regions, there are particular windows where in that 100 base pairs, for example, the X chromosome and the Y chromosome sequences are only 75% identical. So of those 100 base pairs, about 25 of them were different on the Y chromosome than on the X chromosome. And that's a lot of divergence. Remember, I just mentioned that two humans tend to be 99.9% similar. So we would expect normally, for if this was the comparison between the X chromosomes, not an X and a Y, but an X chromosome from one person and an X chromosome from another person, this line would be around 99.9%. So there would basically be none of this structure, valleys, in this plot. But the Y chromosome, because it's very different in sequence from the X chromosome, shows these regions of divergence. And then there are some spots, like this almost 1,000 base pairs of X chromosome sequence. By the way, this is not human X and Y chromosome sequence. I didn't tell you which species this is, but it's not humans. But there's a region in this species of about 1,000 base pairs where there's less than 50% nucleotide similarity between the DNA that's there. So there is DNA here on the X chromosome, and there is DNA there in that 1,000 base pairs that's present on the Y chromosome. But those sequences have mutated one compared to the other so much that you can barely recognize them as being similar sequences. Over half of the sequence is different. And that's true again here, and especially down here at this end of the plot. So that's the basics of what a percent identity or VISTA plot is, and that's how you read it. For each little bit of the chromosome sequence, you can see how similar the two chromosomes that are being compared are. Here it's the X and the Y chromosome from one individual. What we're going to be doing is looking at comparing the same chromosome from two different species. So we'd be looking at, say, chromosome 2 from a bird and chromosome 2 from a lizard, and looking at how similar and how different their chromosome sequences are. Now, here's how this gets back to the concept of looking for regulatory sequences. We know, again, that exons, the parts of transcripts that encode proteins, are very highly conserved. That means they're regions where it's very, very, very unlikely for sequences between even distantly related species to change much, because the amino acid sequence of the protein usually is conserved. The protein sequence needs to be the same. And so we expect that exons, which each of these blue vertical bars represents, the position of an exon of a gene, so the genes are listed here in black, and then their exons in those blue columns, show us where the coding regions of genes are. And so it's not surprising that everywhere we see there's an exon, the percent identity between the chromosomes is a little bit higher, right? All of these valleys that we see within the genes in introns are not in the exons, they're in introns. And where we see this great divergence between the X and the Y chromosome sequences are regions that are not in genes. So the only genes in this region of the chromosome are here. Outside of genes, there's a lot of variation. There's some areas where the chromosomes are very different in sequence. But what happens when we have a region of a chromosome, for example here where I've circled it in yellow, why would that region be highly conserved between the two chromosomes or between two different species if there's no gene there? That is phylogenetic footprinting, and that's how they can be identified using this sort of a plot. It's a region of high conservation, but in a region that there is no known gene. So where there are regions of high conservation of DNA sequence, but it doesn't encode a gene, that makes geneticists suspect, this doesn't prove, that that might be a region that contains a regulatory sequence. And again, the regulatory sequence might be highly conserved because the particular DNA sequence of that region of the chromosome is functional. It may be, for example, has a protein that binds that specific sequence. And so if that protein binding to that regulatory sequence is critical for the organism to survive or to develop or whatever, then natural selection will resist mutations in that region of the chromosome. Anytime there is a mutation, that individual would be less fit and they wouldn't reproduce as much, or they would die or be eaten by a predator or something. And so they wouldn't pass that mutation down to their offspring. So natural selection sort of weeds out individuals that would have mutations in regions A that are exons. And we know where those are because geneticists have located the positions of the genes. And so the regions that we don't know about would be regions that are highly conserved that are not in genes. Those might be what these authors refer to as conserved non-exonic elements. So that should be obvious to understand at this point, not the whole concept necessarily, but the wording. Conserved means the sequences are the same or almost the same between distantly related organisms. And non-exonic element means it's part of the DNA that's not in an exon. And as I just mentioned, that is probably a hallmark of regulatory sequences, that they're conserved and they're not in exons. So this is just to reiterate a point that's already been made. A big question is it mutations in the coding regions, the exons, or is it mutations in sequences that regulate the expression of genes that are more important in the evolution of feathers? And this is beyond just the evolution of feathers. This is a critical question in the study of evolutionary development. Do mutations in protein coding regions of genes or in their regulatory sequences more frequently used to allow the evolution of new traits? Now, despite what I just said, that we're probably more interested in regulatory sequence change, there's a really important concept to understand about how mutations impact protein sequence. So mutations in exons that encode amino acids. So it's important to take a look briefly at the codon table and to remember how the process of translation of messenger RNA works and how mutations impact that process. So first, let's take a look at the sequence on the top, which has 3 codons, or 3 sets of 3 nucleotides. Each codon is read by the ribosome, and then we can use the codon table to understand which amino acid would be encoded by each codon. So AUG, there's a single codon. That's the only codon of the 64 on the table that encodes methionine. And then we've got AUU, which encodes isoleucine. And then we've got GUC that encodes valine, because that's a 3 amino acid exon. And then below, I've indicated what happens if 3 of those different nucleotides become mutated. So we're going to take a quick look at how that influences the translation of the sequence. AUC, AAU, and GUG is now the sequence that's in, let's say, the first one is in species A, and the second one is the same sequence of the same gene from species C, or species X, doesn't matter. So AUG, being the only codon that encodes methionine, means that if any one of those 3 nucleotides is changed, undergoes a mutation, that will certainly change the amino acid that's encoded by that codon. So now we have AUC, which encodes isoleucine. We've got a point mutation that causes an amino acid change. In the second position, we had isoleucine AUU, and now we have a mutation in the second position of the codon, the second amino, or second nucleotide rather, to change to AAU. So where's AAU? It's here, so that encodes asparagine. And then that third mutation, again at the third position in the codon, GUC to GUG. GUG encodes valine just like GUC does. So the point of this is just to remember that the position of a mutation can have an important and powerful impact on the sequence of the amino acid that's encoded by that codon. So the big point here is simply to understand that while mutations are random and can happen to any nucleotide anywhere, exon or not, first position, second position, or third position in an exon, in a codon, there are particular mutations that are more likely to cause amino acid change. And it's the amino acid change that's likely to change the function of a protein because it changes the structure of the protein, the way the protein folds, and therefore the way the protein interacts with other proteins and molecules inside the cell. So some mutations are more likely to be involved in evolution than others. And that brings us to an important point about the neutral theory of molecular evolution. It's pretty straightforward. It says it all here. I'll summarize it for you quickly. Mutations happen regularly and at random. And this theory also suggests that most of the time a mutation happens, its effect is neutral. That means that it doesn't have an effect in the situation of protein coding genes. If a mutation happens, the neutral theory suggests that those mutations are likely to be neutral, to not change the amino acid, and thus not be able to be acted on by natural selection because the protein sequence isn't changed. On the other hand, some mutations in protein coding genes will be what's called a nonsynonymous mutation, which is one of those mutations where a single nucleotide change does change the protein that's encoded, and that amino acid change may influence the structure and the function of the protein and therefore be able to be acted on by natural selection. So a big question in the field of molecular evolution is trying to figure out, just based on sequence data, how can we predict whether a non-synonymous substitution that happens in a gene, again, that's a mutation that changes the amino acid, was beneficial or detrimental? Because Natural selection acts to remove detrimental mutations. If it's a mutation that changes an amino acid and that amino acid change is not useful to the organism, natural selection will act to remove that mutation that's a detrimental mutation. But evolution also requires mutations that are non-synonymous because those are the only ones that change amino acids and so change the structure and the function of a protein. So while some of those mutations are going to be bad changes and get selected against by natural selection, useful mutations that improve the fitness of an individual relative to its other members of the population, those will be selected for. Their offspring that also have that same mutation will be more healthy than the offspring of everybody else in the population, and eventually that mutation could be selected for an increase in frequency in the population. So what we're talking about here is the concept of positive and negative selection. And so these are terms that we're going to hear quite a bit in the study of evolution and development, especially molecular evolution. We have the term positive selection, which is also called diversifying selection. This is what we call a mutation that changes an amino acid and are beneficial. In other words, those are mutations that are non-synonymous mutations. Those are the ones that change the amino acid at one spot in the gene relative to the mutation. Which mutation, what spot of the exon was that mutation in? That changes the amino acid that was encoded by that codon in the messenger RNA. The opposite is what is generally thought of to happen to exons, which is purifying selection or negative selection. And in that case, we expect the amino acid sequence of the protein not to change. But we just saw in the codon table that there are some mutations, the synonymous mutations, where a DNA sequence will change, but the amino acid sequence doesn't. And so those are called synonymous mutations, and those can't be acted on by natural selection. So in the case where the sequence of a gene that encodes a protein is resisting change because any possible change to the amino acid sequence would be bad, the effect of that is if we looked at the DNA sequences of those genes in a bunch of different organisms or a bunch of different species, we would see more synonymous substitutions than we would expect by chance. And remember, that concept of chance is that mutations happen randomly. So mutations don't care which spot in which codon or which nucleotide is there now and which nucleotide gets replaced. Usually, there are some caveats to that broad description I just made. But broadly, that's true. So if mutations are happening randomly, and then they're being tested by natural selection, we can actually look at whether or not we see more synonymous mutations in the absence of those random non-synonymous mutations that would also be happening. So if there's more synonymous than we'd expect, just based on random mutation that happens over time, that suggests that what evolution is doing and natural selection is getting rid of the mutations that are non-synonymous. And if we see more non-synonymous substitutions than we would expect by chance, that suggests that those non-synonymous substitutions are being kept by natural selection because they improve the fitness of the organism. So that's all well and good, but here are two more important and critical caveats. That you can't just sum up how many non-synonymous changes there are and how many synonymous changes there are and do easy math like compare the two numbers. Oh, there were 9 synonymous changes and 1 non-synonymous change. So that must mean that there are very few non-synonymous changes, so this must be purifying selection. And that's because, as we saw in the codon table, and we'll look at again in a minute, different codons have different opportunities to have either synonymous or non-synonymous mutations happen to them. So what we have to do with those sort of raw numbers of just looking at how many non-synonymous mutations there are and how many synonymous mutations there are, we have to normalize those values by the opportunity for mutations to have been synonymous or non-synonymous. So I'll explain that in a little bit more detail. What we're going to be looking at is this really critical concept of measuring positive and negative selection using these values that are referred to as dn and ds. dn is also sometimes called ka, and ds is sometimes called ks. They're used interchangeably. And what dn stands for is it's that same easy thing we could do. We could look at two genes, sequences, and we could count how many non-synonymous substitutions there were just based on the codon table. It's this per non-synonymous site part that's the intricacy. That's the normalization. We have to know not only how many non-synonymous substitutions there were, how many mutations that changed amino acids, we also have to know how many opportunities there were for non-synonymous mutations to happen. Likewise, with DS, that's the S, the synonymous substitutions, that we count up how many non-synonymous mutations we observe, and then we have to divide that by the opportunity for synonymous mutations to have happened. And so these denominators, the number of synonymous and non-synonymous sites, have to be determined individually for each case, for each gene and pair of genes in comparison. So I'm going to show you an example of how to do this. So in this example, let's consider a single codon, AUU. We've already seen this one once. That was isoleucine, AUU. Now let's take a look at that part of the codon table. There are 3 different codons that are related to each other in sequence that encode isoleucine. All 3 of those isoleucine codons start with AU, and then the third nucleotide can be U or C or A. And it's only in that 4th position where if there was a change from U to C, that would be a synonymous mutation. So, right, we have AUU right now. If we change that 3rd mutation, if a mutation happened to happen there, if there was the first opportunity or the first possibility is U becomes C, that would not change the amino acid. Still isoleucine, so that's a synonymous mutation. If that U becomes an A, the second choice, if a U is going to mutate to some other nucleotide, there are only 3 other nucleotides that it can change to. U to A at that 3rd position also encodes isoleucine. But if U becomes G, now we have a non-synonymous mutation where AUG encodes methionine, not isoleucine. So the way to count the number of sites, this is again going to be the denominator for DN and DS, is you take each codon, and so this would be the target species that we're going to compare another sequence against. So we do this just for one of the 2 sequences that are aligned with each other. And in that first position, we say, okay, we're going to focus on the first position, A. What happens if you mutate the A? We're going to count up how many non-synonymous changes there could be and how many synonymous changes there could be. So if A changes to C, that's AUU to CUU. That's a change from isoleucine to leucine. So that's one non-synonymous change. What happens if A becomes U? That would be UUU. That's phenylalanine. So AUU to U, AUU to CUU encodes leucine, AUU to GUU encodes valine. So in that first position of this specific codon, in position 1, there are no possible synonymous mutations. All 3 possible mutations, changing A to one of the other 3 nucleotides, all of those are non-synonymous. And that is what this fraction 3/3 indicates, is that at the first position of that codon, here, remember, we're tallying the number of non-synonymous mutations. That 3, the denominator, indicates, which this is the same number here, indicates that there are 4 nucleotides, and since 1 is present, there are only 3 other possible nucleotides that A could become, G, C, or T. In this case, 3 out of 3 of those are non-synonymous changes. If A changes to T, it's non-synonymous, changes to C is non-synonymous, changes to G is non-synonymous. So 3 out of 3 possible mutations at the first position are non-synonymous. So 3 out of 3. And because there are 3 total nucleotides that it can change to, that's why the sum of these has to be 3. Because we're counting what are the 3 different nucleotides that that mutation could be if there's a point mutation. That's why then there are 0 out of 3 possible synonymous changes. At that first position, there's no synonymous possible change. So you look at the second codon, or the second position of the codon, position 2, that U, so A something U, A UU encodes isoleucine. What are the 3 different nucleotides that could replace that second U? A, C, or G. And that's moving this direction across the codon table. So AUU becomes ACU, mutation at the second position. We go from isoleucine to tyrosine, or sorry, threonine. Mutation of that second nucleotide to A encodes asparagine. Mutation of the second nucleotide in the codon G encodes serine. So once again, any mutation, if we're looking at this codon, the second position, any mutation is going to certainly be non-synonymous. So again, at the second position, we see that non-synonymous opportunities are all 3 of the nucleotides. And there are 0 then opportunities for a synonymous mutation at that nucleotide. So now we're starting to get this idea. We're starting to build this idea that most of the time, mutations at the first position of an amino acid, this is not always true, but first position of a codon rather, most mutations are going to be non-synonymous. Second position, most mutations are going to be non-synonymous, not always, but most of the time, just like in the first position. Third position is the exception to this rule. In the third position, there are a lot more opportunities for synonymous changes. So let's take a look at that. AUU. So again, AUU encodes isoleucine. There are 3 different nucleotides that could replace U in the third position, C, A, and G. So if U mutates to C, that's synonymous, doesn't change the amino acids. That's 1 synonymous change. U to A at the third position also encodes isoleucine, so that's our second synonymous change. But U to G encodes methionine, which is a non-synonymous mutation. So 2 of the 3 possible changes are synonymous, and that's why we add them to the synonymous line here. And 1 of the 3 was non-synonymous. So again, the total for that is 3 different possible nucleotides at position 3. So if we sum across the lines, the first position, the second position, and the third position, we see that for this codon, there are what we call 7/3 non-synonymous sites. We'll abbreviate that, non-synonymous sites. That's my own abbreviation. That's not a standard abbreviation. And relative to that, there are 2/3 synonymous sites. What this means, hopefully, will be intuitive. That's some math, which is important to be able to do, but we can set aside for a second. Intuitively, what this means is that for most amino acids, if you mutate the first position or the second position of the codon, those are going to be non-synonymous changes. And for a lot of the amino acids, if you mutate the third nucleotide, that won't change the amino acid that's encoded. So in general, you would predict that if random mutations are happening, this is why this calculation is important, why I'm saying approximately 2 out of 3 mutations should be non-synonymous changes. And about 1/3 of them, ones that happen at the third nucleus or nucleotide of the codon, should be synonymous on average. But you have to do this calculation for each codon. So each codon has its own unique signature of the number of synonymous and non-synonymous sites. So you could do this for each of the 64 different codons, calculate how many non-synonymous opportunities there are for change and how many synonymous. Now let's see what we do with those information. So let's walk a little bit slowly through this example. First thing to notice, ignore all the table, just focus on what I'm about to tell you. Notice that I have one sequence here, an RNA sequence with 3 codons. So one codon is shaded gray, the next one white, and the third one gray. That is from species A. So that is the nucleotide sequence of 3 codons from species A. And just like we did on the previous slide, I used the codon table to figure out how many synonymous opportunities and non-synonymous opportunities for change there are for each of those 3 codons. And so we just looked at AUU and we looked at AUG. So AUG is unique. Any mutation at any position will change the amino acid encoded. AUG is methionine. Any mutation to that codon anywhere in one of the 3 positions will change methionine to something else. That's not true for isoleucine. There are 2 synonymous changes possible at the 3rd position, and 1 is non-synonymous. And then I just added a 3rd codon here just to increase the complexity of this a little bit for this demonstration. So in this example, GUC, any change at the 1st position would be non-synonymous. If we replace G with A, C, or T, that changes the amino acid. Same for the 2nd position. And this is one of those codons where the 3rd position could be any of the 4 nucleotides. And that's why there are 0 non-synonymous sites. If a mutation happens here, it's guaranteed to be synonymous. All 3 possible changes are synonymous. So if we take a look then at, for this section of this chromosome, from this one species, there are 7 1/3 non-synonymous sites and 1 2/3 synonymous sites, adding across all of those numbers. And so that sums to 9, which is expected because there should be 9 sites because there are 9 nucleotides. So what those 2 numbers give us are the denominator for the DN calculation and the DS calculation. So now let's take a look at actually doing that calculation. Once we have the denominator, which is the number of synonymous sites and the number of non-synonymous sites in this region of the chromosome, we need to compare this sequence to something else. And that's where species B and species C come in. So I've given the sequence of the same region, same part of the same chromosome from species B and from species C. So these are pairwise comparisons. What we do is we take one at a time. In this case, we're comparing species B back to the sequence of species A. So we're going to compare that sequence to that sequence. A and A, no mutation. So we don't record any change. We're just going to classify what those changes do to the protein, the amino acid. So the mutation, if there's no mutation, then that's 0 synonymous and 0 non-synonymous changes. Here we have a U in A and a C in B. And if you go look at the codon table, you'll see that that changes the amino acid, as we already looked at. AUG is the only codon for methionine, so mutation at that position would certainly be a non-synonymous change. And then here, there's also been a mutation from G to C. That also changes. So in this case, we're not looking at the 3 nucleotides together. We're saying for each of these individual nucleotides, there's a C here. So we're saying if in the original species there had been 1 change, just that 1 nucleotide we're looking at, would AUG to AUC have been a synonymous or a non-synonymous change? And AUC is a different amino acid, so that's why it's marked as a synonymous change. In other words, what we're not doing is comparing the codon here to the codon there. We're comparing 1 nucleotide at a time. We're imagining that in species B, if there are multiple changes in species B, we're looking at 1 nucleotide at a time and imagining that the rest of the sequences are the same. That's why for this nucleotide ACC, for the middle C, this first C, we're imagining that that would have been a replacement in this codon, ACG, and that would be non-synonymous. And then again, for that mutation, just that 1 mutation, if we imagine that it had started with AUG and there was a single mutation, that C, we look at its effect. And then we move on to AUU becomes ACA. The 2nd position here was a non-synonymous change that encodes ACU, encodes a different amino acid than AUU. So that single mutation would have been or was a non-synonymous change. And likewise, in this case, A AU single change, A, encodes the same amino acid as AUU, and that's scored as a non-synonymous change. And then we move on to the third codon and do the same thing. There's no mutation in this case in the first position. There's a mutation at the second position, it's non-synonymous. And there's a mutation at the third position, and it's a synonymous change. So we sum up across the entire sequence alignment we're looking at the number of synonymous changes, which is 4, and the number non-synonymous changes, 4, and the number of synonymous changes, which is 2. Here's where we can calculate dn, which is the ratio of the number of synonymous changes to the number of non-synonymous, sites. Remember, non-synonymous sites is that idea that there's a particular opportunity for mutation to cause non-synonymous change. And there are a lot more ways for non-synonymous mutations to happen because most nucleotide sequence changes to an exon, to a codon, would cause a non-synonymous change, would cause a different amino acid to be encoded. And that's reflected in essentially the ratio of the number of non-synonymous sites to synonymous sites itself. There's about 4 times more probability of a mutation being a non-synonymous mutation in these 3 codons than it is to be a synonymous mutation. So that's why we take the number of non-synonymous changes, the numerator, and divide it by the denominator, the number of non-synonymous sites. And then we take the number of synonymous changes we've observed and divide that by its denominator, the number of synonymous sites, the possible number of synonymous mutations. That calculates for us dn, which is whatever 4 divided by 7 1/3 is, and ds, which is 2 divided by 1 2/3. And then we can do the same thing. In this case, I'm looking at a third sequence. Do exactly the same thing with a sequence from a third species. Again, this is a pairwise comparison. So now we're going to look at the sequence of species A. I'm not going to do the full analysis. And compare it back again to species A. 1 nucleotide at a time. So there's a difference here at the first nucleotide. A becomes C. So now we have to ask, is C, UG, is that single change, that C, synonymous or non-synonymous? It's non-synonymous, so we mark it down. And then we move on through the rest of that sequence. And when we're done, we see that in this case, there are 5 non-synonymous changes and 1 synonymous change. And it's important to point out that in species B and C, just for this example, it's important to point out, the total number of mutations between sequence B and sequence A is 6. The number of sequence mutations between sequence C and sequence A is also 6. So if we just sort of blindly counted up the number of mutations, we wouldn't get any useful information out of this because all we would know is, well, there were 6 mutations. But it's where the mutations are and whether or not the effect of those mutations are synonymous or non-synonymous that is important. So now let's take a look at how we interpret those dn and ds values by calculating their ratio, which we call dn-ds. And that's the name of this analysis, or at least colloquially named the dn-ds analysis. So calculating dn-ds is straightforward. You take the value for dn, 4 divided by 7 1/3. That's the numerator, and then you divide it by the denominator, whatever the calculation for ds was. In this case, 2 divided by 1 2/3. And that calculates to 0.46. Likewise, for example C, where we compared this sequence, sequence C, to the sequence of A, we saw that there were 5 non-synonymous changes and 1 non-synonymous change. So these were our dn and ds values, and when you divide dn by ds, you get a value of 1.14. So what does it mean? And that's what we're going to finish up talking about. So again, sequence B differs from sequence A by 6 mutations, more non-synonymous than synonymous. And remember, though, that there is a approximately 4 times greater likelihood that any mutation at random in any, just if I closed my eyes and threw a dart at the DNA, and whatever nucleotide I hit changed to a different nucleotide, there's a probability that on average, I would have 4 times more synonymous changes than synonymous changes, because there are more opportunities to change amino acids than to make a mutation and keep the same amino acid. So this ratio dn to s normalizes for that probability, or that possibility. And this means that there are fewer, because this is less than 1, that means this is a fraction. dn over ds is smaller than 1. That means there are fewer non-synonymous changes. The numerator is small compared to the denominator, the number of synonymous changes. So this gets back to the idea of, well, what's happening in natural selection? In this case, when we see a value of dn ds less than 1, that seems to indicate that we're seeing fewer non-synonymous mutations than we'd expect, which we interpret to mean that when non-synonymous changes do happen, because they will happen because mutation is random and any nucleotide is subject to mutation, when those non-synonymous mutations happen, they don't stick around evolutionarily long enough for them to exist in the population so that we could collect an individual, extract its DNA, and get that DNA sequence. In other words, dn and ds less than 1 suggests that mutations in these codons that we're studying and the lining are bad. They're decreasing the fitness of organisms that get them. We know that they're getting them just because mutation is random, but the ones that do get them are eliminated by natural selection. So the only individuals that stick around in the population for us to study are the ones that have more synonymous mutations than non-synonymous. So even though the absolute value, the number of non-synonymous mutations is still greater than the number of synonymous, that's not important, because there's a 4 times in these 3 codons, it's 4 times more likely for a random mutation to be non-synonymous. Here, we're only seeing a 2-fold increase in non-synonymous mutations over synonymous, which actually means there's a decrease in non-synonymous mutations that stick around through evolutionary time. Certainly, the ratio of mutations when they occurred was absolutely 4-fold, 7 1/3 non-synonymous mutations for every 1 2/3 synonymous mutation. But what's happened again in evolution is most of the individuals that got those non-synonymous mutations weren't as healthy, weren't as fit evolutionarily. And so they didn't pass that mutation on to as many offspring as ones that were getting just synonymous mutations in this gene. On the other hand, C shows us the opposite pattern, which is, even though, again, there were a total of 6 mutations, just like in species B, those were quantitatively or qualitatively different mutations. That even though there was 1 more non-synonymous change and 1 fewer synonymous change, that ratio, comparing that to the number of opportunities for those 2 different changes, gives us a dn/ds value of greater than 1. So what this means is the numerator is bigger than the denominator. The number of synonymous changes is greater than the number of synonymous changes. And so this tells us something kind of odd. And that is that it looks like in this species, in species C, there's a benefit. The individuals that had lots of synonymous mutations, or perhaps just key synonymous mutations that did a particular thing to this protein, those individuals that are getting non-synonymous mutations are more fit. So then that is kind of weird, because normally for most genes, we think, well, the DNA sequence isn't supposed to change because the protein sequence is important. And if the protein sequence changes, then the protein probably won't work as well. And so most genes, in general, it's thought, undergo this sort of selection like species B, this gene, where we see lots of synonymous mutations relative to very few non-synonymous changes. That's evidence that the sequence of this gene is important. So what's going on? What sorts of things and what sorts of genes might we expect to see DNDS values greater than 1, where it looks like after natural selection is finished, the individuals that had more non-synonymous changes, amino acid changes to this protein, are better off, they're more fit than individuals that were getting fewer non-synonymous mutations or only synonymous mutations. It's a little bit counterintuitive. So I want to end with this slide, and then we'll talk about examples of proteins in humans that do undergo that second mode of evolution where you get more non-synonymous mutations than you'd expect by chance. So quickly, to summarize the interpretation, if dn is equal to ds, if the numerator and the denominator are the same, then that ratio is 1. And that tells us that there's no selection. All that's happening is random mutation is occurring, and no mutation is better or worse than another. So that's what happens if the dn-ds ratio is equal to 1. If dn is not equal to ds, that suggests that selection is occurring, that mutations are either better or worse and make the recipient either fitter or less fit. So we can't say for certain yet which type, synonymous or non-synonymous, is beneficial. That's where the value of that ratio, dn to ds, is important. So we just looked at an example where it's greater than 1 and less than 1. All this slide tells you is the vocabulary you need to know. When dn, the number of non-synonymous substitutions per synonymous site, is greater than ds, that makes dn-ds ratio greater than 1. The numerator is bigger than the denominator. That suggests, as I mentioned on the last slide, that there's a benefit of amino acid change in this region of this gene. This is called positive selection or diversifying selection. Natural selection is acting to increase diversity. Somehow, changing amino acids in this part of this protein is better than keeping them the same. The other one that we looked at was dn less than ds, so that ratio would be less than 1. And that is what we probably normally think of as most genes, where the DNA sequence, the gene sequence, shouldn't change because the protein sequence is important, and it shouldn't change because if it did, that would break the function of the protein or cause it to act differently or misbehave. This is called negative or purifying selection. So those are equivalent. We've got negative or positive selection, and we've got purifying or diversifying selection. Those terms can be used interchangeably. And so we saw an example of that purifying selection as well. So to close, here are some genes from a separate paper that were tested and were found to have positive selection, dn-ds greater than 1. More non-synonymous mutations happening relative to the possibility of them happening than synonymous change. So to close, here's what some of these genes do. What sorts of proteins would you imagine would want to undergo, or natural selection would want them to undergo, positive selection, change, protein sequence change? This is bringing up a really critical and basic evolutionary concept that the same DNA sequence can be beneficial or detrimental depending on the environment that sequence and the individual that sequence has is in. So one mutation, non-synonymous or synonymous, doesn't matter. Let's talk about non-synonymous since that's changing amino acid sequence. That mutation could be beneficial in some circumstances, but detrimental in other circumstances. And that's what's similar about a lot of these genes. Not all of them, but a lot of them. ApoBec3G and CCR5 are both proteins that are involved in HIV infection and defense in humans. So it's probably not surprising that since HIV pandemic is, relative to the last few decades, that we've seen an increase recently in positive selection for those proteins. As those protein sequences change and adapt to either avoid, well, not either, to avoid being able to be bound by the HIV virus, That's a benefit of DNA sequence change. Non-synonymous changes the amino acid sequence of these proteins so that the HIV virus can't bind those proteins, because the proteins on our cell surfaces are changing shape, and that's a benefit. Likewise, the next gene, MHC, major histocompatibility complex, our immune system, proteins that we depend on to have different sequences to create different shapes that bind and recognize different antigens, different invaders of our bodies. SRY, the gene that encodes the protein that starts male sex determination in mammals, undergoes positive selection. It changes its sequence more rapidly than other proteins. Likewise, genes that are involved in sperm-egg recognition. Two different species should not, well, by definition, do not reproduce with each other. And this is why. The receptor protein on the egg, zonapaloocytic glycoprotein, and the ligands that bind those egg surface receptors undergo concerted evolution. Their sequence changes, and that sequence change is beneficial because if two different species happen to mate with each other and they create a hybrid, often those hybrids are less healthy. So presumably, the positive selection, the diversifying selection of these proteins is to ensure that matings occur only between individuals of the same species that have the specific protein structure of the sperm, and the egg has the specific protein structure that recognizes only sperm from the same species. So molecular diversity is good in that case. Lactase is another example of recent positive selection in humans. That is evolution near the gene that allows adults in humans to continue producing lactase, the enzyme that digests milk sugars. So some individuals in the population don't have those mutations and let them become lactose tolerant. Most individuals in the world don't have that change and they're lactose intolerant. So that was a protein that underwent selection, positive selection for diversity, when milk from animals became a nutritious opportunity for humans. That is, when humans started herding cows and goats, essentially. When we started consuming milk, that provided a new environment in which it was beneficial to have changes in that protein. I'll be a little bit faster now, I'm running out of time. Melanocortin-1 receptor has everything to do with pigmentation in human skin. So you can imagine that migration of humans around the world, there was different selection for whether or not you produce a lot of pigment or not a lot of pigment. And that can produce different levels of selection inside the coding region of that gene. Senses, olfactory receptors, the proteins that we use to detect smells, and our red and green opsin proteins, proteins that are expressed in the retina that allow us to absorb and detect different colors of light. Super useful proteins to have to allow us to sense our environment. Those have undergone positive selection in humans. FOXP2 is important in speech. Humans speak. We have more complex language than a lot of other animals. Potentially evolution of the FOXP2 gene and that positive selection explains some of that. And then cytochrome C oxidase and ASPM and microcephalin, which are involved in human brain size evolution. Humans have much bigger brains compared to basically any other species in terms of relative size of the brain compared to the body. And those two proteins have undergone positive selection, presumably reflecting that increase in brain size. So that, again, is an overview of molecular evolution, how to detect whether or not genes are undergoing selection for diversity, positive or diversifying selection, or undergoing change, sustaining selection against change, which is purifying or negative selection.